Hello, everyone, and welcome to this Integrated DNA Technologies webinar. Today's webinar is on our Lotus DNA library prep, a true customization for NGS. My name is Ginny, and I will be serving as moderator for today's presentation. Today's presentation will be given by Simon Dunbar, APAC NGS Field Applications Manager. Simon is a member of the IDT NGS Field Applications Group, providing support to researchers using IDT's range of NGS solutions in the field. Simon completed his bachelor's degree in biomedical sciences with honors at the University of Ulster, following that with postgraduate studies at Queen's University, Belfast, where he earned a master's degree in molecular biology. Prior to working at IDT, Simon investigated the therapeutic potential of siRNA for treatment of conditions caused by dominant negative mutations. He has been with IDT for 5 years. Simon's presentation should last around 30 minutes, and following that presentation, he will answer as many questions as possible from attendees. The question and answer session will be conducted by myself. As attendees, you will be muted, but we encourage you to ask questions or make comments at any time during or after the presentation by entering your questions in the Q&A box. Also, please note that you can expand the presentation slide window for easier viewing. In case you need to leave early or want to revisit this webinar, we are recording the presentation and will make the link to the recording available on our website a few days after the presentation. We will also post the recorded presentation on our YouTube and Vimeo channels. You will receive links to these in a follow-up email. So now, let me hand it over to Simon for his presentation.
Thank you, Ginny. Hello, everyone. Thanks for dialing in to today's presentation. I'm excited to give you all a preview of the newest addition to our portfolio of NGS products, the Lotus DNA Library Prep Kit. This kit is intended to provide researchers with a simple, fast, and cost-effective method to construct NGS libraries for many applications. We will begin with a quick overview of a general NGS workflow, followed by a review of the steps involved in the library construction process. The remainder of the webinar will focus on our Lotus DNA Library Prep Kit, looking at the overall workflow and highlighting the flexibility of the kit for multiple applications. Choosing the right adapter for your application is an important part of the library construction process. We will review how to leverage adapter options for your application. Finally, to finish, I will share some data from our R&D groups on some of the applications, mainly whole genome sequencing for PCR-free and PCR-amplified libraries, but also for targeted sequencing data. This is an overview of a general NGS workflow, usually beginning with a nucleic acid sample, which can be DNA or RNA. This nucleic acid sample needs to be made compatible with Illumina technology through the library construction process. From there, you can go straight to whole genome sequencing, or alternatively, you can choose to perform some kind of enrichment for targeted sequencing of only the regions of interest. IDT specializes in targeted sequencing, and we have two technologies that we offer for enrichment. You can either go for amplicon sequencing, shown here in the bottom, and use our RAMP-seq technology, or you can choose to use a hybridization capture approach by using our Xgen range of products in combination with the Lotus DNA Library Prep Kit, shown here on the top blue line. After enrichment, you proceed directly to sequencing, then analysis. No matter which route you choose, IDT has an option to support your research with optimized workflows and reagents. Let's spend a moment to cover the library preparation process before moving on. Library preparation is a series of enzymatic steps that takes long, double-stranded DNA fragments, shortens those through fragmentation, and adds sample indexes and flow cell attachment arms. This can be broken down into 3 main steps. The first step, shown here on the left, involves taking the double-stranded DNA sample, shearing this to shorter fragments, and performing end repair and A-tailing. The result is blunt-ended, short, double-stranded DNA fragments with adenylation marks shown here by the A sign. The next step is ligation of adapters to the adenylated fragments. Adapters are added to allow flow cell attachment, cluster generation, and also sample indexing. We will go into more detail on adapters in a few slides. The final step on the right-hand side is an optional amplification step via PCR using standard P5 and P7 primers. I will show how this general workflow applies to the Lotus DNA library prep kit. Our Lotus kit is modular, and what we mean when we say that is that you can use a single kit for many applications. You can perform PCR-free whole genome sequencing, PCR-amplified whole genome sequencing, or targeted sequencing all with the one kit. We have an efficient workflow which enables a fast turnaround time with minimal tube transfers. Each of the three colored blocks on the right represent the three modules from which the entire workflow is completed. We begin with a single tube fragmentation, end repair, and A-tailing, and because we use an enzymatic shearing method, there is no need for acoustic shearing equipment. There is also time savings associated with not having to manually shear libraries. After this, you proceed to adapter ligation, followed by a cleanup step. Moving on to the second module shown in green, this is an optional library amplification module, and this can be omitted for PCR-free workflows. The third and final module is a cleanup step, only recommended for whole genome sequencing applications, and this step is to remove any excess adapters prior to sequencing. The entire process can be completed in approximately 2 hours depending on which workflow you choose. This is an overview of the Lotus chemistry. In this example, we are beginning with a genomic DNA sample as the input. This undergoes enzymatic prep, which involves fragmentation, end repair, and A-tailing. This step takes around 40 to 60 minutes. After this, you move on to adapter ligation. It is at this point you have to make a choice on which adapter type to use. You can choose to use stubby adapters shown here on the left-hand side, or you can choose to use full-length adapters shown on the right-hand side. I will go into more detail in a minute on this, but this step should take around 20 minutes. After ligation, you move on to an optional PCR step. The time taken for this PCR will vary based on the input amount into your library prep. If you have lower amounts of input, say 1 nanogram or 10 nanogram, you will have to do more PCR cycles than you would if you were to input, say, 100 nanogram. As a guide here, it's good to allow for 10 to 30 minutes to complete this step. Let's look at adapter options and indexing strategies. Adapters are added to samples to allow attachment to the flow cell and enable cluster generation, but also for sample indexing. On the left here, we can see full-length adapters. And let's look at the structure for a moment. At either end, you can see the P5 and P7 sites. These allow for flow cell attachment and cluster generation. Internally, we have the I5 and I7 sample indexes. These are used for demultiplexing your samples, allowing you to run many samples at the same time on the sequencer. When you choose to use full-length adapters, flow cell attachment sites and sample indexes are added during the ligation step. After ligation, you can go directly to sequencing or perform library amplification. This differs from stubby adapters. If you choose to use stubby adapters, flow cell attachment sites and sample indexes are not added during the ligation reaction, but are instead added via an indexing PCR step using indexing primers. PCR is mandatory when using stubby adapters. IDT is the leader in oligosynthesis with over 30 years experience, and we leverage this capability to create high-quality ligation adapters. So when would you use one type of adapter over another? If, for example, you wanted to perform PCR-free library prep, you need to make sure that the sample indexes and flow cell binding sites are added during the ligation reaction. This is why we would recommend full-length adapters for PCR-free workflows. You can choose TrueSeq compatible full-length adapters which contain unique dual indexes, or you can select the XGen dual index UMI adapter, which has a unique molecular identifier in addition to the unique dual indexes. For those interested in low-frequency somatic variant calling, we recommend the UMI approach, as this will allow you to use UMIs to build consensus during variant calling. If you are using targeted sequencing or any PCR-based methods such as metagenomics, you may want to explore the use of stubby adapters. Stubby adapters have a higher ligation efficiency than full-length adapters, which will result in higher yields from your library prep, giving us a more complex sample with a greater representation of the original material. In this table, we have summarized all of the applications that the Lotus DNA Library Prep Kit is compatible with. We have indicated the best adapter choice for each application with a tick mark and highlighted the best alternative with a blue dot. As Janine mentioned earlier, these slides will be available after the presentation, and you can use this table as a reference point for your adapter selection. IDT gives you flexibility in your adapter design, and you can now customize adapters using our brand new Adapter Configurator tool. It's a very simple tool, and by following through the steps seen here on the right-hand side, you can build your own custom NGS adapters. You begin with the library prep method. Is it TrueSeq style or next era style? Then you can choose the attachment mode for TrueSeq style adapters. Is it stubby or full length? Next, you can choose the number of indexes. IDT has up to 3,840 unique dual indexes available. Then you move on, you can choose the amount of material needed. There is an option for the number of reactions or a nanomole amount. It's highly customizable and very easy to use. I'm going to move on and show you some data from our R&D efforts with the Lotus kit. I'm going to begin with some whole genome sequencing data. The Lotus DNA kit can be used for whole genome sequencing applications and is compatible with PCR-free and PCR-amplified workflows. If you want to do a PCR-free workflow, then we would recommend beginning with sufficient starting material. Our recommendation is to use at least 100 nanogram input for PCR-free workflows. As there will be no amplification with this workflow, you need to make sure that you use full-length adapters, either TrueSeq compatible UDIs or XGen UMI adapters. For PCR amplified workflows, you can use any input from the recommended range of 1 to 250 nanograms. If you choose to use full-length adapters, you have the option of performing PCR with P5 and P7 primers to increase your library yield. If you choose to use stubby adapters, you want to add indexes during PCR, so you'll need to pair the stubby adapter with indexing primers. Let's look at some whole genome sequencing library profiles. Here you can see traces for PCR amplified and PCR free libraries. PCR amplified libraries are shown in dark blue and red, while PCR free libraries are shown in green and light blue. A common question we have been asked is, why do my PCR-free libraries run ahead of my PCR-amplified libraries on the bioanalyzer? This is due to the forked nature of the Y-shaped adapter. In PCR-free libraries, the adapter is not completely duplexed and has floppy ends. In PCR-amplified libraries, however, the adapter is duplex and runs at the correct size. When running PCR-free libraries, you will see this profile. You will know that it's not due to underfragmentation, but due to the forked nature of the full-length Y-shaped adapters. PCR-free libraries will always run at a higher molecular weight. So how do the libraries from the previous slide look when you sequence them? Shown here is an example where we took 100 nanogram of cell line DNA and ran it through our PCR-free and PCR-amplified workflows. Shown here is a normalized coverage, and the theoretical limit of 1 is shown with the grey dotted line. You can see that for PCR-free libraries shown in dark blue and PCR-amplified libraries shown in light blue, they line up well with the theoretical results. Below this, you can see a histogram plot, and this shows 100 base pair windows at different GC percentages. This data shows that we are not skewed in either direction towards a particular GC content, indicating that we have uniform coverage with minimal GC bias. We get excellent coverage across our recommended input range, and we wanted to display that, so we chose this experiment. For this experiment, we used the ATCC MSA 1000 standard. This is an artificial microbiome made up of a mix of 10 bacterial strains of varying GC content from 30 to 70%. We constructed NGS libraries from the MSA 1000 standard using the Lotus DNA library prep kit and tested a range of input amounts for library generation. And you can see the results of this experiment here, with dark blue indicating 1 nanogram input, light blue indicating 10 nanograms, and green for 25 nanograms. We are measuring the observed genome coverage shown on the y-axis and comparing that to the expected genome coverage from the ATCC data on the x-axis. We see that there's a strong correlation for expected versus observed, despite having differing input amounts, different size genomes, and varying GC content, indicating that the Lotus library prep kit delivers consistent performance. This makes Lotus an excellent choice for applications with variable GC content. Looking into coverage of varying GC content further, we prepared libraries from 3 standards of bacterial species of varying GC contents. In the top panel, you can see a bacteria that has around a 30% GC-rich genome. In the middle panel is E. coli, which has a balanced genome of around 50% GC. And in the bottom panel is Rhodobacter, which has a very high GC content of around 70%. We are again here looking at normalized coverage, and the theoretical limit of 1 is again indicated by the grey dotted line. The experimental data in blue, green, and orange lines up well with the theoretical limit. We are seeing consistent coverage over a wide range of GC content from 30 to 70% with minimal bias. Moving on, let's look at some data from Lotus for targeted sequencing and show examples of how Lotus fits in the targeted sequencing workflow with our XGen range of hybridization capture products. Often researchers are interested in the protein coding regions of the genome. These next couple of slides will focus on whole exome sequencing. The Lotus library prep kit has been optimized for use with the XGen range of high capture probes and reagents. The addition of Lotus to our product range now means that IDT can provide you with an end-to-end solution for high capture, starting with Lotus DNA library prep kit in combination with IDT's high-quality ligation adapters, and for use with enrichment for IDT XGen lockdown probes and hybridization reagents, and finally onto sequencing. For those who are not familiar with the Hive capture workflow, I'll spend a minute here to give a bit of an overview. Starting at the top here with prepared library fragments. This is a mixture of fragments and will contain our sequences of interest shown in dark grey, but also sequences we are not interested in shown in light grey. The first step in the workflow is to add blocking oligos. Blocking oligos are used to prevent daisy chaining of adapter arms. The end of each library molecule has the same flow cell binding sequences, and in the absence of blocking oligos, these can potentially hive to each other and give us non-specific pull-down, which should be avoided. After we block the adapter arms, we add XGen lockdown probes shown here in light blue. These are 120 base biotinylated DNA probes designed against your target regions of interest. We then use strip-tavidin beads to pull out those target sequences and wash off unbound off-target sequences. A unique feature of IDT lockdown capture probes is that every probe is individually synthesized, QC'd, and normalized prior to entering the pool of probes. This is unique to IDT and not a feature of any array synthesized capture panels. We are commonly asked what are the important metrics to look at when analysing capture data, so we'll cover some of those important metrics now. Uniformity of coverage is an important metric when analysing capture data. In this example of GP9, I have shown the ref-seq representation of the gene. This is the coding portion shown in dark blue, and this is the read pileup you see with the IDT exome panel. In an ideal scenario, you would have coverage of the target exon, and it would be perfectly flat. You would just cover everything to whatever your mean coverage is and no more. And as you see, we do a pretty good job of that. To represent this on a more global level, rather than just a single gene, we can look at the percentage of reads, which is a metric many of you may be familiar with. This is really the distribution of coverage. Say your coverage is 45x, you would want 100% of your reads to be right there. How tight the distribution is important, but also the position. So with the same amount of sequencing, you would like this to be as far to the right as possible, giving you the most coverage. Another way to represent this is the percentage of bases at greater than X coverage. Here you want to see a step function with 100% of your bases covered at your mean, and then you want it to go to 0 and no extra coverage. So you're looking for as vertical a line as possible and as far to the right as possible. So what does this data look like for IDT Lotus in combination with our IDT Exome? Here we are showing data from libraries made from 100 nanogram of cell line DNA with stubby adapters and UDI indexing primers. This was captured with the IDT XGen exome panel and sequenced with 50 million reads per sample. Looking at bases covered at greater than X on the Y-axis versus coverage on the X-axis, we can see a nice step function which uniformly drops off. A sequencing depth of 20x is commonly used as a minimum requirement for viable calls. This is indicated here on this data set with the grey dotted line. And as you can see, 97.2% of the target bases are covered to at least 20x with 50 million reads. An issue a lot of customers face when performing capture experiments is the ability to adequately capture GC-rich regions. This is a problem as GC-rich regions of the genome are often important to the researcher's questions. This study back in 2006 showed that no matter the length of the exon, whether they are short, average, or long exons, it appears that first exons will always have a higher GC content than internal exons. Knowing this, we thought to look at capture performance of first exons as a benchmark for capture. The example shown here is RB1, and we chose RB1 as an example because, as you can see, exon 1 has a very high GC content, while exon 2 is a more average middle-of-the-road GC content. You can see the read pileup for both exons 1 and exons 2 here, and you can see that both exons are covered extremely well with no discrepancy of coverage between either. So this would not require any extra sequencing power. Looking back again at the example of GP9, which I had shown earlier, we can see that this is a region of the genome that also has a very high GC content. However, again, there is uniform coverage when using the Lotus and IDT XGen exome combination. The last example of GC-rich capture that I want to show is concerning APOE. APOE is poorly represented in current NGS datasets. You can see the data below for APOE from the Broad Institute's EXAC consortium. The data we see here is the average of thousands of samples that have been through exome sequencing. As you can see, APOE has 4 exons, and there's a dropout region between exons 3 and exons 4. To make up for this, it's not uncommon for people to supplement exome sequencing data with whole genome sequencing data. This is a costly exercise. If we look at APOE data from the IDT Lotus plus exome combination, we can see end-to-end gene coverage with all 4 exons covered well and no dropout between exons 3 and exons 4. This data would not require supplementation with whole genome sequencing data. The difference in performance can be attributed in part to the ability of the Lotus DNA library prep kit to handle varying levels of GC well. To summarize, the Lotus DNA library prep kit offers flexibility in its workflow, allowing you to use a single kit for many different applications. Lotus kits can be paired with ligation adapters of your choice. You can use our recommended adapters for each application, or you can customize your own adapters using our new adapter configurator tool. The Lotus DNA library prep kit provides uniform GC coverage for whole genome sequencing of PCR-free and PCR-amplified libraries. We have demonstrated consistent performance across the input range, shown how Lotus fits with IDT's Exchange range of capture products to provide an end-to-end capture workflow. This will enable you to get good coverage across regions which have been traditionally difficult for capture. Thank you all very much for attending today's webinar. I'm happy to answer any questions that you may have.
Yes, thank you, Simon, for the informative presentation. If you have a question and have not done so already, please type it into the Q&A box. Okay, there are a couple of questions for you here, Simon. Let me get this out first. I'm pushing it out to the slide area. Oh, this is, okay. There's a question here, and it asks, what is the advantage of using RAM-seq versus target capture enrichment?
Sure, that's a great question. So capture, so using XGen capture is better suited to larger spaces and lower frequency variants, because when we use capture, we have the ability with ligation-based adapters to incorporate UMIs, so we can drill down to a greater level of sensitivity. Whereas when we're using amplicon sequencing, we haven't yet found a way to incorporate UMIs. So you can't use those to build consensus and drill down to low frequency variants. And also, in its first iteration, RAM-seq is currently only for short indels and hotspots, whereas we can tile across entire regions with hybrid capture.
Thank you, Simon. We have another one coming in. Let me push this out to the slide area so that everyone can see the question. Right. Is this library compatible with the Agilent Bates system?
Yeah, absolutely. You can use the Lotus Library Prep Kit in combination with the SureSelect Bates. That's no problem. It's obviously optimized for use with our XGen hybridization capture probes, but certainly will work with SureSelect also.
Thank you, Simon. Here's another one. It's for those who are with automation. Wonderful. So this question comes in. Do you have automation protocol for the Lotus kit?
Not yet is the answer to that question. So it's a really new kit to us. We've just launched in the past sort of 8 or 9 weeks. So at this stage, we don't have automation available, but that's something that we hope to address in the future. And we can take that offline if the customer has a particular instrument that they're interested in automating on, and we can see what we can do.
Okay. Here's another question that came in is regarding PCR, 2-step or 1-step. Here we have it. Let me push it out. For everyone to say 2 steps or 1-step PCR, which is better for NGS?
Sorry, I don't quite understand the question.
Let's put this to later, but there's a new question that came up. I'm pushing out the slide area. When and where this recorded webinar will be available. As moderator, I'll answer that. It would be available perhaps on Monday. Yes. Any further questions from the audience? Okay, that is all the time we have for questions. I want to thank all of you for attending today's presentation. I also would like to thank Simon for his informative presentation, as well as the question and answer session. This is one of a series of webinars we are presenting on NGS, as well as other topics. We will email you about these future webinars as they are scheduled. Also, as a reminder, a recording of this webinar will be posted shortly on our website and at www.youtube.com/IDT-DNA bio, B-I-O, all right? And over there, at that channel, at that YouTube channel, you will also find several other educational webinars on many other topics, such as next-generation sequencing, genotyping, qPCR, and general molecular biology. Thank you again for attending, and we wish you the best of success in your research. Oh, there are several more questions. Simon?
Yes.
Simon, there's several more questions.
Okay, I see there are. Sorry,
go ahead,
several
more questions. Yeah. Let me push one out here. Specifically on GC-rich regions. Is there a special protocol for capturing GC-rich regions?
There's no special protocol that we use for capturing GC-rich regions with our exome. I guess with the IDT capture protocol, it's just important to maintain the temperature during the wash steps to ensure great capture of GC-rich regions. That's probably the most important step in our protocol, particularly for GC-rich regions. If we have the wash buffers too high or too low, that will impact on the capture of those regions.
Okay, thank you, Simon. We have one more question, just one more question. This question is pertaining to adapters, whether our adapters are compatible with Illumina and can be used for the same run.
Yes, so all of our adapters are Illumina adapters. We are authorized resellers of the Illumina indexes. So we have the 8-base and 10-base Illumina indexes available to us. But in addition to that, there are a limited number of indexes available from Illumina. I think the maximum is 384. So with sequencers having higher capacities these days, such as NovaSeq, you really want to maximize the space on your flow cells. So we have in-house barcodes indexes. So IDT has up to 3,840 unique dual indexes. And they are compatible with the Illumina indexes as well.
Thank you, Simon. Thank you for that answer. Right. Thank you all. I believe that's all the time we have for today. As mentioned earlier, please visit our YouTube channel for further videos. The recording of this presentation will be, and the link for it will be sent to you in a follow-up email. Thank you all for attending. Wish you the best for your research, and goodbye.